Okay, welcome. Here we go with lecture 5. We're going to learn more about image processing as it pertains to microscopy, and this is Dr. Giorgi. The topics we will cover this week are how to do overlays and what they are, how to properly do a contrast stretch, how to do gamma, and we're going to look at how to use ImageJ. So we're going to start with overlays. And what are they? Well, we're going to skip ahead one slide so I can tell you about them, because this semester is a digital semester or a online semester. We're doing things in a different order. And at this point in the class, we usually go specimen prep, image acquisition, and then post-image acquisition processing, step 3, which is where we're here. We're actually starting this semester with image processing, and then you're going to get to do specimen prep and also image acquisition when you get the scopes at home. Okay, so in fluorescence microscopy, the way it works is that you hit your specimen with light of a certain color, high intensity light of a particular color. It absorbs that light and then it gives it back to you, but as a different color. And we will cover this in great depth after this module. We're going to go into fluorescence for a few weeks, but that's the gist of it. And so when you're doing fluorescence microscopy, color is really important. Color is your signal, you might think, right? Because our eyes see color and we react to it, we get all happy, we're like, oh, I love color. But your signal, in truth, is just the fact that there is something coming from that part of your specimen. Because the point of fluorescence microscopy is it's very specific, and you can have a lot of specimen and only part of it will react when you shine light on it. So this is an example from a paper by a phenomenal microscopist and scientist, Ron Vale, over at UCSF. He's also started a company or two. So in this paper, they're looking at immune system cells, and they're looking at 2 cells. So one cell is presenting its find, let's say it's find, it's known as an antigen. It's basically a cell trying to tell another cell, hey, look what I found roaming around the body. Do you think we should do something about it, like attack and destroy it? Say, for instance, I don't know, a virus that you might have found that wasn't there and shouldn't be there. Okay, so what we're looking at here are 2 cells. This first cell on the top, and then this second cell on the bottom. They're both quite round. Immune system cells tend not to have a lot of morphology, in other words, shape, morphology, morphology. So it's fancy science word for shape. They're just round little blobs. You can't tell one apart from another. Oh, wait, you can if you use fluorescence microscopy, because as you can see, one of these cells reacts when hit with, here, let's look at the overlay, hit with red light, and the other one, well, it's hit with red to give green. But anyway, one of these cells is a different color than the other cell when you look at it under fluorescence microscopy. And this is the image you will generally see. And this image is full of info because they're actually, so this is one set of cells, and this is another set of cells. And what they're showing here, let's just look at the overlay in this paper, is in this case, in this scenario, these 2 cells, they're different. I can tell them apart. I can tell a green red cell from a blue cell, and they've got different functions in the immune system. You can read the label below and ask me more about it. But these 2 cells are doing 2 very different things. One's a T cell, one's an antigen presenting cell. And then they're messing with these cells. They've got this system to look at these 2 cells and how do they interact. And when they mess with them by changing something genetic, in this case in the cell, they see that this protein appears right at the at where the two cells interact, whereas it doesn't here. So this is the info contained in this image, is like, look, in this case, there's just the two cells touching each other. In this case, the two cells touch each other, and there's also this protein right there where they touch each other, nowhere else. So exciting finding. How did they do it? They did it with fluorescent microscopy by labeling everything with different colors. How did they actually image it? They took images one color at a time. So that's what known as one channel. So the red channel, the green channel, let's see, this is probably green channel, red channel, blue channel. And because they know how to do microscopy, they actually took those images with a black and white camera, because a black and white camera is more sensitive. If you can have a color camera, color CCD chip in your camera on your microscope, but it actually cuts down a third of the light, because it's basically putting, it's called a Bayer mask. It's putting a colored filter in front, and you're basically, the bottom line is you're losing light if you go for color. And you don't need light, you just need, is there a signal, and how much is there, and where is it? And you know that this black and white image was taken using your green channel. Why do you know that? Because you kept track, because you're really good with the GLP, the good lab practices, and you keep track in your image log in your notebook. You keep track in the file name. You just keep track is the really important part of this. And so you get a black and white image out of your scope, and that's okay. You know that in real life it was giving you bright green. And then you can go back and apply a lookup table, and look, it's bright green. Again, same thing for the red channel, same thing for the blue channel. And then this is an overlay. So there were 3 images, and here they are, 1, 2, 3. So green, red, and blue. By the way, it's a usual order in which you present the images. Just for convenience, we always present the images as green, red, and then blue. And then you present an overlay, which is, I can tell that this is different than this, but it's just nice to see them together. It just gives my eye more info. But if I put them all together as black and white images, I'm not going to be able to tell what's what. So, okay, let's add the color back in. And besides, we love color, so why not? So all 4 of these images is actually an image, okay? In microscopy, when we say image, it can loosely mean just this overlay. It's also known as a composite. So you've overlaid 3 different images in this case. Sometimes you can add in a bright field image, like a DIC image, and that's 4. It's an overlay of 4 images. Or it can be an overlay of 2 images. Maybe you only had 2 channels, 2 colors. Whatever is appropriate for your image. The overlay is you overlaying several images, or making a composite of several images and turning it into one image. But when I say, give me the image of the T cells, you're going to give me this. You're going to give me all 4. You're going to show me channel 1, channel 2, channel 3, and then the overlay. And that's what you're going to get to do in lab. And in general, image, know that it has a broad meaning. It could be just this panel of your figure, or it can be this thing, it can be this whole thing. Image can be many things, basically. It's a loose word, just like saying bright field. It can mean many things. And note that sometimes people will present these parts of their figure, parts of their image, or sub-images of their image, however you want to think about it. They will present these in color too. It's kind of a microscopist expert thing to just be like, I know better. I'm going to present the individual channels in black and white because there's more contrast. Your eye can see what's going on better. But then I'm going to give you the color because, of course, the overlay has got to be in color. Here's another image right below. So I was talking, if you're not seeing my mouse, I was talking about the top 4 squares or rectangles that are labeled as control images, right? And this, in this paper, they nicely call it overlays, the fourth one. So that's part of why I chose this. And then the bottom 4 images that make up one image, because there's one set of info that's coming out of all 4 of those. It's the same thing. It's the same channels. It's also this is the name of the protein that they're actually looking at, but notice they've put the green, red, and blue little boxes around it, so you can kind of be reminded that although you're seeing it in black and white, it was actually in real life green, red, or blue. And here you go back with the color. You might be saying, where's the blue? That is, I love this image too, because it's so dim, it's there. You might have to, depending how it goes, by the time it gets to your screen, you might not see it, but there's a second cell here that's round, and it's just very dim because real biology is like that, especially when you're doing real experiments with cool stuff. Sometimes you can barely see your signal. In fluorescence, it's always about, almost always about, can I see my signal? It's only plants where you're like, whoa, too much signal. But other than that, you're always trying to get to see something very dim and very ephemeral. That just means it comes and it goes. This is another reason why, like, if you ever by accident should purchase or have to use a color camera in fluorescence, you'll soon be like, oh no, why didn't I buy the black and white camera? Everything's going to be much brighter. It's just going to be a better camera. So again, this was green. No, yes, this was green, and this is what they saw in the green channel. This is what they saw in the red channel. It was green in real life, but they captured it as black and white and then added back the green color back here. It's kind of teal because it overlaps with the other colors a little bit. So it's actually overlapping with the blue too. So that's why you see it as a nice teal color. All right, so going back, when I say image, when anybody in microscopy says image, it might be just literally one square or one rectangle or something like that, or it might be a grid. It might be multiple images of the same thing that all add up together to give you a certain piece of information. So it's green, red, blue, and then the overlay, all 4 of those together are one image. Okay, so overlay or composite, you can put them in a grid like this, or you can just put them in a row like Ron Vale and his lab did on this paper, as you wish, but you want to present them together. So either this little grid or, you know, a longer grid basically. So square, rectangle, either way is good. But the point is you have each channel, preferably as a black and white image. It's okay if you've colored it back with the lookup table, but you probably acquired it as a black and white image if it was fluorescence in a fluorescence scope. And then you do the lookup table to get the nice color for the overlay, and you merge 3 channels. You're going to do this in lab next week. Okay, on to the next topic, contrast stretch. So as you know well by now, this is showing you a lookup table, the standard linear lookup table at a 45-degree angle, where 1 is 1, 2 is 2, 3 is 3, 4 is 4, and so on. It's probably what you thought in your mind was happening to all your images until you learned better. At least I did, until I thought about it. I was like, why wouldn't you just show an image like the image is? Well, nothing is really shown as the image is. Everything needs to be decided upon and shown. Every time you take a digital image and show it on a screen somewhere, something is being decided about it and probably changed. So let's play with the lookup table. Now we know the lookup table exists and is at the heart of displaying your images. Let's play with that. Let's take that lookup table and play with it. And I did this using Fiji in my video that I've also linked on this page. In my video from lab, and we'll do more, I mean, from the live lecture last week, and we'll also do more when hopefully I'm able to broadcast from lab this coming week. So if you take that lookup table and you move the minimum max, you can end up getting a really steep slope, like I've shown here under the word max. Max means there's the maximum contrast, because you get a lot of contrast if your slope is that steep. You might start to get too much contrast and lose some resolution. So, oh, it's so funny sometimes. You haven't had what we usually do as lab one. And in lab one, which you will do later when you have the scopes at home, a big theme of this semester is that everything is a trade-off in microscopy, probably in life too, but I'll let you decide that. But in microscopy, everything is a trade-off. And so we know that microscopes exist to give us contrast and resolution. Well, the truth is you have to choose a bit between those two, and the more contrast you get, the less resolution, and the more resolution, the less contrast. And you'll see that in very real ways when you get your scope at home, because you'll play with it and it'll become kind of visceral. It'll become part of your brain and your hands. When you're working on a microscope, it's something very fundamental that you always know, is you're always making a trade-off. Do you want to see more stuff? Do you want to see it better? Everything's a choice, and everything's kind of a binary choice between two good things, and you can't have both of them at the same time. And that's where the art of being a really good microscopist comes in, and where the more experience you have, the better you get at it. And why computers and automated microscopy are, they exist, and they're helpful and useful and getting better all the time, but there's still a lot of work for the rest of us that aren't computers. Okay, but know how to use computers. So we're not computers, but we use them and we're good. Okay, so anyway, there's a trade-off. You can have a lot of contrast, but you're gonna lose your resolution if you make your image too contrasty. There's more contrast than the 45 degrees. That's probably a nice contrast depending on your image. You can also end up changing the lookup table to have less contrast. There's no real reason to do this in microscopy. There is when you're on the scope, as you'll see when you're using something called the iris diaphragm. But when you're just sitting at the computer, I doubt you're going to choose to give it less contrast. You never know, though. And then I think I showed you in the live video that you can flatline it. Just play around with it so there's no contrast. Obviously, that's not useful. You just wiped out your image. But play around with this on Fiji with the images you have. Grab the lookup table and mess with it until you have that in your gut understanding of what the lookup table is doing to your image or how it's displaying it. So again, we normally use a linear lookup table. Well, we use that as the starting point for thinking about it, and also hopefully your image comes off of the scope that way, unless you've told it otherwise. So the linear lookup table is this 45-degree lookup table. This is just showing some example data. So this is what you'll see on the software. There's a histogram drawn and your lookup table is over it. Note that the values on the Y and X axis are actually different. It's actually two charts on top of each other, okay? So for your data, on the X axis, it's gray levels. On a Y axis, it's pixels, number of pixels that have that gray level. For your lookup table, and this is what I've drawn because we're interested in the lookup table right now, it's the gray level also, and on the X axis, and on the Y axis, it's still the gray level. Only, this is where it gets confusing, only this is the gray level you're going to display it as. So On the x-axis, it's the original gray level. On the y-axis, it's the one that you're going to display it as on the computer or on the phone. Okay, so that was your classic lookup table, nice linear, 0, 0, 1 is 1, 2 is 2, etc. But what if your data looks like this? First of all, don't let your data look like this. Don't let your histogram look like this. Don't let your histogram grow up to be looking like this, or a cowboy, sorry, old country song. Okay, so, and bad joke, by the way. Okay, so what if your histogram looks like this? You have not maximized your dynamic range, but sometimes you're just not able to. You had to go fast or whatever, something happened. So it would have been nice if you gave it a little more exposure, got a better image to start with, but this is what you have. Okay, fine, let's fix it using the lookup table. Let's do some post-acquisition image processing. So after you've acquired the image, you're going to make a copy of it, and now you're going to process it. So here is a very commonly used trick in image processing that you're just going to do so much once you realize you can do this. It's called a contrast stretch. So what you're doing is anything in the black levels, the dark black, dark gray levels that you didn't use, you're just sort of lumping them in and calling them all 0. And really, in this case, 2 is the new 0, right? You're just saying, if it's 0, make it 0. If it's 1, you know what, make it 0. If it's 2, let's call 2 0. Okay, after 2, we'll start acknowledging that, you know, these are different gray levels, but anything 2 and below, it's 0. That's also known as the offset value. And it's a legit thing, especially because when you get to the fancier scopes, you're going to learn about, there's all kinds of noise that comes in. So in anything where you're looking at data, there's signal and there's noise. So there's what's real and what you're trying to see. And then there's just what random things pop up because there's electricity going through your system. And the good thing is that the noise fluctuates. And a lot of times, there's actually 7 kinds of noise just when you're doing digital imaging. So stick with us if you want to know. We'll cover it in the advanced classes. But one of the ways that we sort of make images better is we take an image of something totally black where there should be no signal and figure out what the noise is and then subtract that, because noise is usually dark and it's random. So anyway, altering the offset is a common thing. And you sort of figure it out on your scope and your system. And then you sort of know, but like I said, you leave some room. You don't stretch out your histogram till it's touching the edges. You're leaving a little bit of space there in case maybe there's some surprises and you want to be able to capture them as you're imaging. So that's the black end. So that's the one end of this contrast stretch. We can also mess with the other end, the top values or the highest values in the gray levels. In this case, I'm only taking you to 5 gray levels. And I'm sort of showing you, oh my gosh, 4.5 and above. We're calling it all 5. So we're throwing away any data on both ends because we're really confident that there isn't data. There's only data where there's signal. And I'm going to use this new Z-shaped lookup table as my lookup table. And what I've just done is called a contrast stretch. And by the way, it is still technically linear with that same looseness, although we call that original 45-degree lookup table the linear lookup table. This contrast stretch lookup table is still linear because there's really one slope. There's one line, one slope. And in fact, let's go back a second. When I showed you this, when I did max and more, those are contrast stretches. Okay, so those are both things. And the one where it says more is maybe something you'll typically end up doing. So this is the original lookup table in red. And now this is the contrast stretch lookup table. It's still a linear lookup table, but we call it contrast stretch. So it's a contrast stretch. Contrast is the key word. We're getting more contrast out of this. Okay, so again, play with this on Fiji and don't believe me, do it on Fiji and learn and see. So you're using a new lookup table to increase contrast because you didn't use your full dynamic range. And so you're kind of compensating for that post-acquisition. Oh, technically you can do this pre-acquisition too. If for some reason you're in some great hurry and you're like, I can't figure out the proper exposure, I can't make this brighter, or for other reasons that might come up, you can do this while you're at the scope using the software that you're using to acquire the image too. Either way, note it in your image log, say that you did that. However, know that it's a legit manipulation for scientific images. The main thing is that you're doing something to all of the pixels in your image, and you're doing the same thing to all of the pixels in your image. And if you can't do the same thing to all of the pixels in your image, that's too bad. You have to. What some people do is take a different image of the part that requires some sort of different manipulation. But you cannot pick and choose. We're going to talk about this a little bit more later. And I may have said this, like if something drops into the middle of your image, you're stuck with it. You can't just Photoshop, make it disappear. You have to either use that image or find a different part of your specimen that doesn't have a bit of cotton that flew in or something like that. Cotton fluoresces a lot, especially the really bleached cotton that is on our t-shirts and lab coats. Okay, so in software, again, this is what you will see. You'll see your data, in other words, the histogram of your image, and then you'll see a lookup table. All imaging software will have this display somewhere. Sometimes they're smart enough to put it front and center. Sometimes you got to go digging through the software, but it's there, and you need it both when you are acquiring the image to make sure that you're getting the full dynamic range and not saturating the ends. And you need it when you're processing the image later on the computer, not on the scope, on a different computer. You're processing your images. You're going to want to see the histogram plus the lookup table. So these are your two great friends, basically. And in the software, you can usually just pick the ends and move them back and forth. And as I said, so this is a contrast stretch, the same exact thing as I just showed you a few slides ago, and anything from that value forward, anything in the super flat, no contrast section, right? Anything in the super flat thing, it's anything before this first pixel where you're acknowledging, this is the start of the gray levels. You've made this the new black. So anything beyond this is also just black, and they're all the same. And anything here is your highest numbers of gray level, and all of these are the same. Okay. So why do we call it a stretch? A lot of people are like, what do you mean a stretch? I feel like I just moved the parameters in. I just made things smaller, right? Like look, it's smaller. Yeah, it looks like that, but what you've actually done is taken all your data and spread it out more because you weren't using the full dynamic range. And by throwing out these end values that you're not using, you're saying, I was using 50 to 200 out of 0 to 255. So now I'm actually going to take those same data points and stretch them out over and pretend that there's 255 gray levels in there. Not pretend, we're going to assign those same data points to 255 different gray levels, or 4096, or whatever 16 bits is. But you're sort of stretching your data to use all the possible gray levels, and this will increase the contrast. And you can take a really bad image and try to really severely contrast stretch it. Like this is a good example of a bad image. There's hardly any gray levels in the original image. And after you contrast stretch it, you you will get a disturbing image. I'm gonna try to show you some in lab, I mean, in the lecture section this coming week. You can kind of look at the histogram there and go like, I bet this is gonna be a very weird histogram. You develop an eye for things that are overly contrast stretched, basically. You're like, they really jump from like there's no continuity of gray levels in this image. It just jumps and jumps and jumps. And in biology, most things, there's a continuity to the grays. And so you see a smooth histogram. If you see a choppy histogram, you're like, yeah, somebody over manipulated this, or maybe contrast stretched it and then got onto a different computer and contrast stretched it more. And it's a mess. Always keep copies before, always make a copy before you do anything. So like I said before, people will say linear lookup table when they mean the 45-degree original 1 is 1, 2 is 2 lookup table, but you still have even a linear lookup table when you contrast stretch, right? You just thrown out the ends basically and stretched your data out back over the full dynamic range. There is such a thing as a nonlinear lookup table. And that is known as doing as gamma. So gamma is the midpoint of the lookup table. And you can actually legit move the gamma, and it's still considered, like I said, legitimate science manipulation of images, because again, you're doing it to the whole image, to every pixel in that image. You're applying the same lookup table to every pixel. So this time your lookup table is not a straight line, but it's a curve. You can use a curved lookup table. And as long as you write it down, and unfortunately editors won't ask you for it yet, but sometimes they do. But keep track of this in your image log. When you do a gamma lookup table, it can be amazing. It can be so, so, so useful. What it does is kind of compensate for images where there's like extreme bright and extreme darkness and you can't see the dark and without saturating out the bright, and or maybe it depends on your image, but this one, for instance, emphasizes the bright pixels. And what it will display it as a curve, but what you're really doing is two linear slopes. By pulling the midpoint down like you did, you get an area with a shallow slope and less contrast, and an area with steeper slope and more contrast. Again, just play with this till it becomes kind of second nature, and you can look at an image and you go, oh, I know, if I contrast stretch and gamma, that is going to be gorgeous. You're going to see everything you need to see. And that's when, you earn the big bucks, so to speak. You become the magician in the lab that people are like, tell me how to use a microscope, first of all, how to prepare the specimens, how to use a microscope. And I got this precious, precious image, but I can barely see anything. How can I see it and still be, what's the legitimate manipulation? I don't want to invent data. And you're like, let me show you how to gamma. And then, you help the world. You help discovery, you help cure cancer. Okay, so gamma is, people just say I did a gamma on it, or you just write gamma. That's the what you saw earlier is the Greek letter for it. The gamma is the midpoint of the lookup table, and you move it up or down so that the lookup table now has two slopes, and it helps when you have extremely bright and extremely dim data points and you want to see them both. It's accepted. Another way that's becoming accepted that you might know from your phones is to take two images with different exposures and then sort of mathematically merge them. And I think your phones call that, what is it, HDR or something like that. And sometimes people do that. It's just the software is a little trickier to do that unless you're using a confocal. Gammas on every software that you're going to use, it's easier, but it's kind of the same thing. It's a bit what you're doing. is you're saying this part is going to have a different, well, you're saying this part of the image is going to have a little different lookup table than this one, but we're doing it to every pixel equally. So when I say part of the image, I mean these gray levels versus those gray levels. And it's really useful when you're doing fluorescence images. It's kind of pointless when you're doing bright field because everything is pretty bright in bright field. But again, in fluorescence, where you might have something really bright in just one part, you know, and then something super dim but super interesting, then you definitely want to check out using gamma. And like I said, keep track of it in your metadata, in your imaging notebook. Some computer software will keep track of the changes you've made, but most of it doesn't. And even if it keeps track, it only keeps track until you save and close it out, and then you reopen it and it's like, what? I have no idea. This is the first time I've ever seen this image. So this is why for now, the imaging log is really important. Don't trust the software unless you know for sure that it keeps track of everything for you. And even then, just get in the habit of putting everything in your imaging log. It only takes a second. So like I said, the imaging software won't keep track of these changes, or if it does, it's only a temporary thing. And always be doing these manipulations on a copy of your original image. So if you go down a weird road, just throw it out and, you know, go to your original, make a copy. Most people I know actually have the original. They're kind of in a sealed vault in 3 places. And then they have the copy that's like the stuff they'll work from. And then if they want to make a change, they make a copy of the copy. And then now you play on that. So, you know, when it's data, when you've sweated for 5 years to get an image that could, you know, open up like the secrets of the universe, if you get the proper image, you're going to get really careful about having copies of copies of copies of that image and not just trust it to one location. So Photoshop does keep track of changes and all of that. It's wonderful for that, but it's also mostly we don't teach it to you because it's expensive, and we're trying to change. We're trying to work on that. If you are working in a professional lab, they'll probably have a license. But to be honest, it's good. It's good to know how to do these things on Fiji because everybody has Fiji. And there are a lot of really great lab situations where they're doing good work, but they just don't have money for a license. Or in our case, we don't have the way to pay them every month. So that's what's holding us up. But if you have Photoshop, you can have fun with it in Photoshop too. And just be careful because 99% of what Photoshop does is not legit for science. But of course, if you can do gamma in Photoshop, that's fine too. All right, last but not least today, Image J, also known as Fiji or Fiji, known as Image J, whatever. So I wrote this when it was Image J, but it's kind of the same in Fiji. Fiji just has more plugins, more macros. It's a bunch of programmers who took the original Image J and said, we're going to put it up and call it Fiji and have a lot of macros with it. So now everybody kind of just goes to Fiji because it's Image J plus some extra stuff. And Fiji literally stands for Fiji is just Image J. So it's just silliness, but it's open source software and it's free from the government, and you already downloaded it for lab. So you already know that. It's your tax dollars that paid for it. Now, it's very simple looking, but it's very, very, very powerful. And it can do a ton of things. And unfortunately, it's built as a menu of all possibilities, equally weighted. So they don't really have buttons up front that are like here's the 5 things you'll always do every single time you open up Image J. Wouldn't that be nice? And then, you know, that would be user friendly. But, you know, if they had some big buttons that just said, look, if you're in doubt, you don't know, just do these 5 things. Everybody needs to do these things. It's not at all like that. There's the things you do every single time thrown in equally somewhere in there with things that you'll never do. Nobody knows what they are, plus a lot of random things that might be wonderful, but who knows, or are super advanced. So this is just taking you through, I think this next year, next week, you're gonna do this part where you do the RGB merge, which is how to do an overlay. So you have to open all the images you wish to merge first, and then go into image, color, RGB merge, and make sure that you put your black and white image that is the green one into the green channel, or into red, and blue into blue. So make sure that you have labeled them well. And in this case, you're using some images that Dr. Mintz acquired for you to do at home. And this is just how to do the composite or the overlay, which you will be doing in lab soon. And this just shows you the drop down. These are your choices. Pretty straightforward. This is actually a saturated bad image that I keep meaning to change every time I get this lecture, but oh well. So there's your composite. Oh, for a second, let's think about colocalization while you're doing composites and overlays. The point is to see, where do things colocalize? Where are they in the same spot? Because what you're getting out of your image are two things. One is, how bright are things, which is very tricky to keep track of, but like, is there a signal at all? I guess is the easier question. And then where is the signal? So is, like, those two immune system cells, is the signal, just where they touch, or is it all throughout the cell? Where is this particular protein I'm looking at? Where is it found? And therefore, that's a huge clue as to what it's doing. In your cell, proteins really do things by interacting with each other. So there's a lot of metabolism that's about, is it near enough? Is it open for business, so to speak? Are things next to each other? I feel like we're learning that as humans now with the viral spread, like, oh, one thing that happens when we're all moving around next to each other is viruses can spread. In cells, everything's moving around, and proteins everything's moving, basically. We're seeing that with fluorescence microscopy, except what doesn't move are cell walls, which is an image. The image that you're looking at is an image of plant tissue, and the cell walls are stained. So anyway, the question often is, with fluorescence microscopy, is it in the same place? Was it always there? Did it move there? But, is it there? And when we do an overlay, there's colors that come from two signals being in the exact same location in your image. And that's known as colocalization. And if a red and green are in the same place, you get yellow. If red, green, and blue are in the same place, you get white. You can see that in this image, which is mostly blue, but occasionally there's all three colors, and occasionally there's just the two. You saw in the previous image, the one with the immune system cells, that you get teal when you get blue and green in the same spot. So we look for colors in these overlays. We're like, yellow, yes, okay, good, that's what I wanted, and so on. But there's a very common error that even a lot of scientists make, because they're just so excited. They see yellow and they're like, yay, the two things are next to each other. That means they can interact. That means my life's work is not wasted. Hooray. Well, you got to think about resolution. And your specimen has 3 dimensions, 4 if you include time, if it's alive and changing. But let's stay with the first 3. X, Y, and Z. So X and Y are in the sort of horizontal flat plane, and Z is what you're focusing. What you're doing when you're focusing is you're moving things in and out of the Z, right? So Z is the 3rd plane. And when you say that something's colocalizing, like in this image where the white parts are, you're saying like all 3 of those molecules that are giving me the color, known as fluorophores, as you'll find out soon, but all 3 of those molecules that are giving me signal, they're in the same place. Yes, they're in the same place in X and Y. Like they're there, they're not a few feet to the left or right. But if you think about the Z, let's go, um, they might not be in the same place in Z. This is kind of like, if you remember my sort of story about Martians looking at us for contrast and resolution. So if we're in a classroom and the Martians are looking down at us from Mars and they're like, ooh, can I resolve how many individual people are there? I think there's one person in that classroom. Turns out it's 31. You know, their resolution isn't good enough. They also want to be able to tell if we're all on the same Z level. And if I were kind of just trying to be energetic and crazy, which I might do in real life, and got up on the desk, and many teachers actually use this trick to get your attention. Anyway, if I got up on a chair or a desk, I am now higher in Z. I'm not in your same Z if you're just sitting down. So if I jump up high or if I swing from the rafters, I haven't done that yet, but you know, it could be that I'm hanging onto the top of the room and you're sitting down normally on the, you know, as a normal person on the floor of the room. Yeah, we're both in the room. The Martian's going to think that we're in the same Z level unless they have a confocal, which is able to figure out these things. So there's two questions. Like, for instance, I might be hanging from the, you know, the top of the from the ceiling. I might be hanging right over you, but we're not actually the same person. There's two of us, me at the top and you sitting very politely on the in your desk. And so to a microscope, to some Martian microscopes, it might look like we're the same person, or that I'm standing right next to you at the desk talking to you, but I'm not. I'm hanging from above, and then I'm gonna, you know, stealth mode, jump down, and I don't know, do some cool sci-fi kind of stuff. Okay, so let's bring it back to actual cells. So here's a cell with a nucleus in gray, and this is what it looks like from the top view, which is what you're doing as a microscopist. You're going to see yellow because you're going to see two molecules that are just right there, sort of in the neck of the extension there of the cell. But reality is that cell is three-dimensional. It's not flat. And the green molecule is up near the top, hanging onto the ceiling, if you will, of the cell. And the red is near the bottom of the cell. Um, and they're nowhere near close to each other in terms of, you know, size and cells. And this is very far away from each other. They're clear across the cell. But you're sitting there going, oh, yellow, I see yellow, because they're actually both in the same location. The answer is no. They're in the same what is called voxel. So a voxel, coming back to this here, a voxel is a 3D pixel. A pixel is when you're thinking about the little square on your CCD camera chip that is capturing the light. That's 2 dimensions, but that light is coming off a little part of your specimen, but it's coming off of the part of your specimen that is closest to you and also the part that's furthest away from you. And this is a profound thing, actually. You'll explore it in your first at-home lab, and you'll also explore it hopefully later on if you stick with us and do confocal microscopy. It's a profound thing to have resolution, not just in X and Y, but also in Z. And can we tell whether things are in the same location in all directions? There's so much information in that, especially since for biology, things that are next to each other are frequently actually interacting with each other and doing something really useful. And if they're not next to each other, they have to interact through a series of intermediary molecules. So if they're far apart from each other, it's harder to see how they're interacting. Okay, so don't ever make the mistake of forgetting about the third dimension, not just X and Y, but also Z. More on ImageJ, just quickly. So you can have these, they can be a little reference book. This is a leaf from cohort 1 and just showing you where to look for certain things. So brightness and contrast. Now there's videos probably that are better than this. And this is, I showed you in class too, this is where you find your lookup table and your histogram. Look, nothing on you who was good at microscopy. I just picked a bad image because it was more fun to do things with it. So you can see it's a very dark image, right? And definitely this image doesn't use the full dynamic range. And you can always try auto. Usually almost everything automatic on imaging software is not satisfactory. Weirdly enough, you'd think it'd be easy for them to figure it out, but it's not that good. We're usually better. It's one of those, I hope I'm wrong soon. I think I will be wrong in a year or two, but until then, don't trust the computers. Use the computers. Your brain is still better for the moment. It can show you the actual values if you want to look at that. That's really cool. You can reset is really important. You saw me do that. Once you like it, save it. Otherwise, it's only changing the display. I like to look at image properties. Sometimes there's good info in there, sometimes not much. It depends on how much it was inputted and how much the software that was used to acquire the image, how much, this is the metadata basically. So sometimes there's some, sometimes there's almost none. And info is the kind of stuff that it can figure out about the image by itself. Basically, it's reading the image file and it's going like there's one channel, etc. Slices and stacks refer to the confocal when you are doing literally image slices. So there's what's known as a Z-stack, where you just go down, you change the focus and take another image and change the focus and take another image and so on. So open, open next, open samples, open recent, and revert are under file usually, and they're all very useful. Let me see if I went, no. Just open is open your, look for your file that you want to process, your image. And open next, if your image is 001, it will just open 002 with the same name. So that's really helpful. And samples are images they put in there that aren't that great, by the way. I showed you some, but they're there for you to play with. Open recent is anything you've done recently. Often that's very useful. And revert is changing back. It is good and bad. It's just changing back to everything, to undoing all your changes. So you can edit, invert. Invert implies an inverted lookup table, which is going to be weird if you have it already in color, but play with it. It'll turn black, white, and white, black. And sometimes it's really entertaining and interesting. Sometimes it's really puzzling, depending on what you're looking at. So like with color, it just gets weird, basically. But as black and white, sometimes you can see things better, or something will pop out and you're like, wait, I didn't notice that until I looked at it under invert. So under adjust, you find display the lookup table. Under color, you find overlays. This is somewhat true and also somewhat different for Fiji, but just keep looking, basically. I actually really encourage you to be a little bit lost, or actually to be a lot lost with software, because all imaging software is different and they keep updating it. It's really good not to be scared of it and just to feel completely free to wade in and just wander around and be like, what does this do? What does this do? I have no idea what this does, but my image looks better. Let me find out, or whoa, that made my image really weird. Okay, I must stay away from that. We're going to talk about analyzing your data a little bit more honestly next semester, but oh yeah, there's histogram. You can find things in multiple places in most imaging software. A scale bar. Scale bars are great if you have done what's known as calibrating your scope. If you told your scope, at 10x, you take an image of actually just a ruler that's meant for scopes and say like from here to here is a micron and so on. And it's nice to have scale bars to just know how big things are. So what else? Again, the histogram, you can find it under analyze. And you see at the bottom of the histogram, there's list and log. And list will give you, oh, I didn't show you, but it'll give you the actual values of that histogram. It'll give you that table that you're doing this week, an assignment lookup table, and log, it just changes the scale, and count is your mean, your minimum value, your max value. Remember, it's a grid of numbers. And so there's a lot of data in, counting the numbers and then telling you what's the top value, what's the middle value, what's the standard deviation. That's really meaningless in this image, but there might be an image where it's useful. So don't be scared. Like, these are programmers and who do this, and they're like, all the possible things we can do, we're going to throw it into the software. Then you have to sort of dive in and be like, I'm going to ignore 90% of this, but this 10% is incredibly useful. And oh yeah, I did show you the list. So I love seeing the list because I'm like, see, there's the table, there's the histogram. There are 0 pixels at 0 value and count. So gray level and how many. There are no pixels of value 1. The first pixel starts with gray level 4, and there's 2 of them. And then, gray level 5, there's 4,337. Thank God I didn't have to count those by hand, right? This is what we got computers to do. I mean, look at gray level 33 has 29,423 pixels. Good Lord. That's a lot of pixels if we had to count that. But this is why we invented computers. Analyze does a lot of really fun advanced things to count things for you and so on. Just, something we'll get into next semester. And you can have a histogram of just the line to see, like, you can look at this and you can see where, the beginning of the line, there's a brightness. That's that first yellow ring, saturated, by the way. Then it gets dim, and then, it gets bright again. If you're doing real analysis of actual data, this gets very exciting, that you can be like, let me count this part of the image. Let me see what's going on with this part. There's even stuff like 3D plots of intensities. I've never seen anybody use it or come up with a use for it, but it's fun to look at. It's just like, there's a 3D version of where things are brightest and dimmest. And okay, well, that was it. Again, over engineering with a computer, you have this, you have the list of things you can do at the very top of your screen when you're using Fiji or ImageJ, and then you have this floating toolbar that usually comes up where you can draw things. It's really a draw things on my scope kind of toolbar, including text or arrows or things like that. And then it also takes you to macros, or things that people just recorded, or plugins, or basically fancier macros, in my mind anyway, to all these other, if you're into software, developing software, you can get lost for an entire 10 careers just on this. And they're always looking for people, by the way. So there's just lots of fancy stuff. I've talked about how there's all sorts of fun lookup tables you can apply that are just random, have no science value, but do them, they're fun. Just so you get a sense of what a lookup table is doing. You can even scribble all over your image. Why would you? Really, this is to draw lines or circle things. But, if you do that, and then you might, it's like revert. Yeah, revert. Okay, that is it for today.